CMorales, Bio 225, June 11th, 2018, Kirby Bauer diffusion test. I have already labeled my agar plate. Now I'm going to carefully drop 8 drops of my onto my broth onto the center of my agar plate. But for this, I'm going to use my S. epidermis nutrient broth culture that has already been active. And I'm going to pipe it 0.5. And I'm going to add 8 drops into the center of my agar plate. Okay. Now I'm going to grab my L-shaped spreader, and I'm going to spread it evenly, making sure to touch only along the straight end. So now I'm just making sure that all of the epidermis is spread on evenly. So now that has a clean layer of broth. Okay, now that is finished, I'm going to go ahead and replace my lid on my agar plate. And then I have already sterilized the lip of my culture vial. So now I'm going to remain this plate undisturbed for approximately 1 to 2 hours until the broth is absorbed by the agar plate. And once it has been dissolved, I'm going to continue on to the next step. I'm now going to begin transferring the proper discs onto the section of my agar plate. I'm going to begin with my penicillin. So I have my tweezers here. I'm going to begin by placing this carefully onto the PNC section. Then I'm going to press it down gently to make sure that it is in proper contact. Okay. And I'm going to return my lid and place my tweezers in my cup of alcohol for 30 seconds. I'm going to repeat this exercise for the remaining 2 antibiotic discs. The next one is going to be the gentamicin disc. Again, making my tweezers are completely dry. I'm going to go ahead and grab the disc and place it into its proper section of my agar plate. And then pressing it down gently, making sure that it is in proper place. Close my lid. Now lastly, I'm going to place my Novobiocin disc into its proper section. Pressing it down gently, making sure that it is completely in place. And placing my lid back on. Now I'm going to allow my agar plate to remain lid side up for about 10 minutes so that the disc can attach securely to my agar surface. Then after the 10 minutes, I'm going to place my agar slides in my incubation location for 12 hours. My Kirby Bauer diffusion has been in my incubation environment for 24 hours. I checked it after 12 hours and I felt that it needed to be incubated for an extra 12, so I now have my agar plate from 24 hours. The Novobicin was the most effective antibiotic because there was almost no bacterial growth near the disc, and the other two antibiotic discs have a type of growth with the S epidermis, but the gentamicin has slightly less growth near its disc, while the penicillin disc was completely covered, which is a little harder to see on the camera.